everybody. Dear ladies and gentlemen, I would like to welcome you to another edition of our BioM Lunchtalk. As we have also English-speaking reference today, the Lunchtalk will be in English. My name is Andreas Berghammer, and I'm responsible for services, cooperation, and sponsoring at BioM. As most of you know, BioM is the cluster management and cluster development company for biotechnology in Bavaria. Today, we are continuing our series on AI ML for biotechnology with two more company presentations. AI has incredibly rapidly become the most dominant topic in many areas of our lives. With this series, we would like to introduce you to interesting companies and organizations that develop or apply AI solutions for biotechnology and can thus contribute to the growth of our industry. Today, we welcome representatives from NVIDIA, a globally active US company that has set standards with its computer chips, especially in the graphics sector, and from Applied AI, an initiative that emerged here in Munich at Unternehmertum a few years ago, is growing rapidly and has recently started supporting other companies in their AI transformation. Before we start, some remarks on the format. As always, we will first have a presentation of about 20 minutes. Afterwards, we will have a few minutes for questions, which we will read out anonymously from the question chat for data protection reasons. You are therefore welcome to post your questions already during the presentation. This will be followed by the second presentation, same mode again with direct questions afterwards, and finally a round with all speakers where you can ask questions on both presentations or on overarching topics. So please participate actively in this event. We will start now with NVIDIA. NVIDIA was founded in 1993, is listed on the NASDAQ and one of the largest companies by market capitalization, and of course one of the largest companies for graphics, developing graphic design processors and chipsets. Our speaker today is Dr. Christian Dallago. He's a computer scientist and bioinformatician working on the modeling of biological mechanisms, for example, the design of proteins for therapeutic and industrial applications using large language models and generative AI. The title of therefore is Large Language Models and Generative AI for Life Sciences. Christian, we are looking forward to your presentation.
Thank you very much. Let me start by sharing the screen. Okay. All right. You should be seeing the screen, I assume.
Yes.
Perfect. Okay. Thanks for the nice introduction. I'm gonna get to interesting, what NVIDIA is in a couple of slides, but I want to start with biosciences meets machine learning. I think about myself more of a scientist than anything else most of the time, although, when you are in industry and in Europe, you kind of have to put on a lot of different hats, as you say in German. But again, science to me is really the driver of what I want to be doing at NVIDIA and what a lot of colleagues I work with are doing. And then, the next topic there is what is NVIDIA building? And apologize, I'll start by my timer so I know how far I am. What NVIDIA is building in terms of products, specifically going into one, which is called BioNeMo. So just touching over it very briefly. And then how do we engage with the community? Obviously, we're not a drug discovery company. We're not a healthcare company. We're building those products for healthcare and drug discovery companies to, do the things that they know how to do best. And we want to be able to interface with them to understand the complexities of how that of those developments and build solutions for them to potentially do things more efficiently, more at scale, etc. So let's start with the actual science part. I come from a lab that was doing computational biology, specifically on proteins and protein sequences. As a matter of fact, the PI of the lab, Burkhard Rost, in 1992, proposed a paper that was combining machine learning neural networks with protein sequences to predict the secondary structure of proteins. Here we see the 3D structure of proteins, but back then That was obviously a very difficult problem. As a matter of fact, we've only been able to kind of solve the problem with AlphaFold 2 about 3 years ago. But in 1992, there were already the first attempts, again, by Burkert, leading some of those efforts in building machine learning systems to learn biology and predict aspects of biology that are interesting. And clearly, these are interesting aspects, right? I mean, we don't see these things except if you have really powerful microscopes, but they make part of us and they make us function and in ways that are fundamental. I mean, here we see hemoglobin for the transport of oxygen, clearly quite fundamental. Alcohol dehydrogenase maybe less so, but still, all of us probably, especially in Munich, are happy after Oktoberfest that we don't wake up with too bad of a headache, and we have to thank biology for that. And this is probably the fundamental slide of my talk, especially as it concerns science. And what I want to convey here is the idea that really what we observe in biology are objects, right? Proteins, genomes, or in chemistry, small molecules. And we have ways of representing, of capturing these objects in digital forms. So that could be the string of amino acids, of residues that build a particular protein sequence. It could be the actual structure of the protein in 3D. So I have the 3D coordinates of the protein, of which we see a 2D projection on the bottom left. And this can be similar, very similar to actual 3D objects in real life, apples, right? So we have a way of encoding an apple in text form, a red apple. We have a way of encoding it in a picture. We can take pictures of apples on the street. We can build models of it graphically by, for instance, having some pixel art here on the bottom right. But what you cannot convey with images or text is the taste of an apple, right? And this is quite interesting, right? But you have an internal mental model that when I tell you red apple, you kind of know what it does. You know what it maybe tastes like. You have a memory of it. So you have an internal brain model of what this 3D object, this physical thing, is in real space. And we want to be able to achieve a similar model of the real world with biology, because we can now represent real objects also computationally. We know that we can generate images today. We can generate text with GPT-like models or ChatGPT. And probably we can do the same thing with biology, especially if there's a lot of data out there to do it. And clearly, because of the cost of genome sequencing going down significantly over the last couple of years, we do have a lot of data that we can leverage to understand the principles of biology by the measurement that the measurements that we can make, which are, for instance, sequence, single cell expression, et cetera, et cetera. And one of the directions in which the lab I was part of at TUM before we were looking at was the comparison to the natural, if you want, the natural language processing. Again, I have a problem with the natural part of it because proteins are also natural language, but we take inspiration from natural language processing, and you can see there are these really big systems, transformer, large language models, that basically just learn by taking a sentence, masking out words in a sentence, and then trying to learn a way of reconstructing the sentence without knowing those words, just from the context of what the rest of the sentence is like. This is similar to how GPT and ChatGPT are trained, the foundational layer of it. And we thought, cool, let's take the same concept and apply it to protein sequences. We have protein sequences, they have a meaning. The order in which the amino acids appear is important. The amino acids that appear in particular positions are really important. So what if we just take that and we start masking random amino acids in the whole sequence, and then we ask a model, a really large transformer model, a really large language model, to just tell us what it believes that the residue at that position should be. And you do this over and over for millions of sequences using, again, very large compute and very large parameterized language models. And you get to some place where the model appears to have an internal understanding, a world model of the connections between amino acids in the protein sequence, the weights of those connections, and possibly also connections of connections. So some higher order information about what makes that protein sequence exactly that protein sequence. You can view this as a similarity to natural language, for instance, by thinking about grammar, right? We're learning the grammar of what makes a sequence a sequence. And grammar is very abstract. It's not relevant to this particular sequence, right? The way subject and object are related to each other is something fairly universal. But then there are also information that are very unique to this particular sequence. And we believe that these models, these large language models, have the ability to learn many of those patterns and encode them in the internal weights of the transformer model. And these internal weights are nothing else than matrices and vectors. So if I take a protein sequence, I can represent it as a vector using a large language model. Very simple. It's a mathematical object. I can compute on it. I can compute on biology. So I can start asking questions like, are these two vectors similar? Or are these two vectors dissimilar? And so I can start thinking about functional annotation, right? Is the function of this protein the same as the protein that I already know the function of? So I can start, just based on vectors and similarity or distance, start assigning some functional classifications, all the way to actually doing structure prediction, real 2D structure prediction the way AlphaFold 2 does, using large language models instead of the system that has been built for 30 years to do the predictions that are now being done with AlphaFold 2. And mind that, of course, the prediction ability of these models in the 3D space, for instance, is not comparable to AlphaFold 2 yet. But the difference is that we started developing these systems in 2019, and concepts like AlphaFold 2 have been developed for 30 years. So I think there's a lot of value in trying to develop these methods further to more align them on what we know or how we know that the systems should be and exist in the biological context. And we've done a lot of work in predicting proteins, in understanding biology, but I think that the future of scientific innovation in the space of healthcare, in the space of materials, is really programmable biology and programmable chemistry. So it's not just being able to, we use the term generative AI quite frequently. It's not just able to generate stuff, it's able to program it the way we want, as we see, for instance, in the case of generating images. And so I'm going to just focus on two topics very quickly here. The first one being protein sequences. And to make the case of protein sequences, I'm actually going again back into the real world. So today we have these systems like Midjourney, Stable Diffusion that allow us to generate images, right? And so the way it works is you go from a noisy state into a state of order, where the order is something that we believe makes sense, and that's an image. It's a complete image, high resolution, et cetera, et cetera. But these systems per se are fairly useless, right? You can just generate images, but what's the point of that? You want to be able to program the image. And how do you program the image? Well, you add an embedding, and an embedding is nothing more than the vector representation of the text that you input into the system. So similar, as I was telling you before, that with language models for proteins, you have vectors that represent the protein sequence. For text, we can represent the vector of the text. And so we just input it into the noise. And then step by step, we start resolving that noisy state into an ordered state, which has that embedding encoded into the image. And so that's the way we get to conditional generation, to programmable generation of images using diffusion-based approaches. And the same diffusion-based approaches are possible in sequence-based and in the biological space. And so specifically here, what we see is a diffusion-based model for sequences. That's what's happening at the bottom. The top is just a validation. It's just showing us that what we are generating with the diffusion model actually falls into a plausible protein structure. So you can basically ignore the top from a perspective of what this model actually does. And it's quite interesting. We can take the model, input a sequence length, a protein sequence length, and it will start resolving some of the amino acids here and there up until the point it has a complete sequence that it believes follows the distribution of a sequence of a normal protein sequence. Now, clearly, that's not entirely useful. As I said, we want to be able to do programmable biology. And a very easy way in which we can condition the generation of sequences is by taking things that we know and we want to conserve. So in this particular case, we start with the green part, which is a functional motif from an existing protein. It is a binding motif. And we just say, okay, we want to conserve these amino acids in a generated sequence. So we input them in a new sequence that the diffusion model has to produce. And then we start populating the various residues that the sequence needs to have in order to be a complete sequence. We fold it, and we see that the folded structure, again, does conserve that binding domain. But still, the rest of the sequence, the rest of the structure are fairly different. So probably it has different properties, new biology that we might be able to explore. And this is in the protein sequences space, but we can go all the way to chemistry and even into chemistry that isn't sequence-based, but it's structure-based. And so specifically, I talk about bound structures here. And one interesting example here is, okay, we have a protein, we have a therapeutic target, and we want to develop a drug that, inhibits or acts, perturbs that particular protein or that particular binding pocket. And so what can we do? Well, now we have a diffusion model that just giving the description of the pocket that we want to target will start generating a conformer, so a small molecule drug that binds into that pocket. This is quite exciting. It's a fairly novel capability, right? We are unlocking new things that we didn't know we could do at the speed or at the accuracy or in the way we are starting to do them now. And all of this is happening since a year or a year and a half. Language models for proteins, maybe it's been 4 years, 2019, 5. But for some of these approaches, we were talking about months or years at tops. And it's been recently shown that this model actually doesn't perform quite as well as we would expect it to, but that's completely okay. It's about the capability, right? It's not about the accuracy at this point. It's just about unlocking new capabilities, in my opinion. And we will work on the accuracy as we have better data sets, better communication between industry and academics, et cetera, et cetera. Again, this field is so new, so we cannot expect it to be perfect from day one. And now to switch gears on, what NVIDIA is doing in the space and specifically why BioNeMo. NVIDIA has been introduced as a graphics card producer. I think that was true until 15 years ago. That's still the way we are perceived, but in reality, we're a platform company. So today, you don't think about the graphics card anymore when you're actually doing machine learning at this scale. You're thinking about high-performance computing systems, right? So you want to think of scale here. The compute unit is not anymore the GPU. It's, if anything, it's a combination of GPUs, so that could be one node. But most likely than not, it's actually a superpod or something really big with a lot of different GPUs and nodes and connection and communication inside of that. And then on top of all of that, there's a software layer for which we have various products that address many of the challenges that you see in genomics, computational biology, medical imaging, federated learning, hospital care. So we really want to be able to address many of the, not as a competition, but just to deliver tools to people to build many of these, really interesting applications using the full NVIDIA platform stack. And so here, we talk about NVIDIA Parabricks. That is a solution for genomic reconstruction, for instance. So if you have your NGS reads and you want to align them to a reference genome to build the genome of that particular sample of that particular individual, you can do that using Parabricks on the entire stack. We have BioNeMo, which I'm going to talk about in a second, MONAI for medical imaging. This is a consortium-led effort. So it's quite, academic and industry-driven. NVIDIA Flare for federated learning, which obviously in the context of healthcare and privacy regulations is quite important. So you want to be able to build systems that are privacy conserving. And then we have NeMo, which is the general, let's say, natural language objects in the real world type of framework that you can use to build, for instance, NLP systems in the clinical context. We have some examples of that as well. And all of this is based on the, built on the NVIDIA platform. And why specifically, so where did BioNeMo start and why large language models? Well, large language models are incredibly complex systems. You need, a lot of hardware and software solutions to actually train a large language model. It's a very computer sciencey, I would say, thing to do. And we wanted to build a system that allows you to focus on the science and on the problem rather than focusing on all of the optimizations, parameters, and all of the things that you usually have to keep in mind for to build a large language model in the case of proteins and chemistry. And so this is where BioNeMo framework comes in handy. It has a lot of capabilities and functionalities for you to just build a large language model for biology and chemistry, not having to worry too much about all of the software stack that you need to build, and it's optimized and scales well for the NVIDIA platform, which could be a single node, it could be the high performance computing cluster that you have available. And on top of that, a lot of people are actually not really interested in just training a large language model from scratch. They just want to use it and apply it for their own problem. And so what you want to do is you want to take a learned model, like the ones that I talked about earlier, like Proc-5, and you want to fine tune it for the data sets, the proprietary data sets that you have in-house to make predictions about protein stability, better antibody production, anything that is more relevant to your use case. And so the framework really covers the entire area from producing a large language model to consuming a large language model to customizing a large language models in the context of chemistry and biology. And we're extending this also to other models, for instance, AlphaFold 2, OpenFold, and diffusion models in the space that are becoming quite relevant, as I discussed about earlier. Okay, and another product line within the BioNeMo umbrella is the cloud APIs. And this has to do with the fact that inferencing is quite complex as well. So if you want to use the models to predict something or to generate something, what you will find often is happening is that setting up the entire infrastructure for inference is again a software engineering complex effort. But also, how do you set up the infrastructure? What nodes do you take? How do you make this efficient and so on? And so with the BioNeMo Cloud APIs, what we primarily built is an infrastructure platform. So we built a system for you to deploy your large language models or your generative AI models for chemistry and biology. And then we also offer many of the open source leading models in this space. So ESM, MegaMolBART, GPT, AlphaFold, OpenFold. So we want to be able to help you jumpstart building applications for drug discovery or for whatever biological chemical process you have in mind without having to worry to set up the infrastructure for it. And then if you come up with an application or a foundational model, like we're featuring foundational models, we're not featuring applications. But if you want to build a foundational model or an application, we want to be able to support you to have an inferencing stack that is purpose-built for that. So you can come back to us and say, hey, I really want this model for internal consumption. I want this model for external consumption for my customers, and we can help you set that up accordingly. And so you can go out and try BioNeMo, the chemistry model on Launchpad. That's a free experience. You can get the framework now. It's available on NGC. It's a Docker container. You can request a proof of concept of DGX Cloud, supercomputer resources, to basically try and BioNeMo, for instance, for 2 weeks on a really large compute infrastructure. And you can sign up for better access to the BioNeMo Cloud APIs today. And then I have about 30 seconds left, but I'm going to very quickly go into how does industry apply AI in biology. And I just make 3 use cases that we worked on with some of our partners. I'm going to talk about InstaDeep in a second more in detail, but interestingly, we really want to understand the leading concepts and problems in the space. So for instance, if you think about proteins in biology, 2 of the really big objects that people are concerned about are enzymes and antibodies. And so we wanted to understand, by collaborating with enzyme developing companies, antibody companies, how can we better our stack so that you can do the things that you do really well, better, faster, more efficiently. And so we built the BioNeMo training service around, for instance, enzyme optimization. And we built some parts of the BioNeMo Cloud API stack around antibody and how to, for instance, predict structures in the antibody space. And now lastly, as I said, I want to focus just 1 second on one collaboration with InstaDeep. And for me, this was really an effort to understand biology fundamentally. It's not about the drug discovery use case, which we covered in this work as well, but it was more to understand biology. Because once you understand biology, you can unlock a lot of different use cases. Again, having this world representation of what biology is. And so here, what we worked with InstaDeep was a nucleotide transformer. So it's a transformer trained on nucleotide sequences. It's not a gene transformer. It's not a genome transformer. It is purposely a transformer that just takes nucleotide sequences. We do not know if that's a coding sequence, if it is a regulatory part of the sequence. We just want to get an understanding of what makes a nucleotide sequence a nucleotide sequence. We do that by putting in a lot of different nucleotide sequences from many different organisms. And we train a very large language model, a 2.5 billion parameter model on those sequences. And what we get at the end of that is this model that actually has the ability to predict promoter, enhancer regions, many of the regulatory elements that are interesting downstream for biology. And from a computational complexity perspective, these models are not trivial. As I said, the computing unit is not anymore the GPU. It's a high performance computing system. So for this model, it took about 1 month to train on 16 DGX nodes, which is equivalent to 128 A100 80 gig systems that are highly efficiently interconnected to each other. And again, I'm already 2 minutes over time. I think the slides will be made available, but there's some useful resources and support material here. We have GTC coming up in Santa Clara in about a month and a half. So if you have or actually it's a month, if you happen to be there, come by and say hi. We have developer ecosystem programs. If you need to, if you are a startup, there's an Inception program. If you need specific trainings, we have Deep Learning Institute. There's a lot of different kits that you can make use of. There's a lot of different developer resources out there. So if you want to develop systems, models, built on the hardware, all of that's available. And if you're an academic and you need some teaching resources for your students and you want to get closer to machine learning or artificial intelligence, there are really interesting kits there. So the Deep Learning Institute teaching kits, you can just get this kit and build some of the material for your own lectures, for instance, for your students based on the kit that is available from NVIDIA. And with that, sorry, I took 3 minutes more than I was supposed to. Thank you.
Thank you very much. No, that's fine. Sometimes a bit too much computer for me, but a very interesting presentation. Questions, please. So one thing, how can existing bio large language models be customized for particular use cases? You said a little bit already in your presentation. How can I adapt the solutions you have?
Yes. So one of the ways is to, for instance, take something like the BioNeMo framework, and you have your own data set. Maybe I go back to the slide just to have the graphic. You have your own data set on your own cluster, and you want to specialize that model for antibody sequences, and you have an antibody data set. So you can use the BioNeMo framework. It will allow you to basically take a trained model, PROT5, ESM2, and then just put your sequences in a FASTA format, which is what people usually in protein biology know, and then just start training or fine-tuning the model for that particular use case. And then you can take that fine-tuned model and build a predictor on top of that. So for instance, in the antibody case, maybe you're interested in if this antibody is soluble or not. And so you can put that on top of the model, all through the facilities which are available here. And the other way of doing that, which doesn't involve training at all, is to use the APIs. So you have your Pandas notebook, and you already have some system for which you predict antibody stability again, for example. And instead of using the features that you extract yourself, you just input the embeddings that come from the API, and then you see, is my model getting better or not? And that's a very, I would say, cheap way of doing it.
So in the beginning, you compared your model against AlphaFold. Can you say something? What's the advantage or disadvantage from one or the other?
That's a very interesting point. So the way AlphaFold is built is by mostly leveraging a concept that is called evolutionary information. So you're not inputting only the sequence that you're interested in the AlphaFold 2 system. You're inputting the sequence and all of the sequences that are related to it that you can find in a database that builds the evolutionary information. And then AlphaFold makes a prediction for that. Language models do not have that requirement, so they have already an encoding of evolutionary information, is my belief, without needing to specifically try to find related sequences. So that's one of the differences that I see. Secondarily, AlphaFold was specifically trained on structure prediction. Language models never had the goal of structure prediction. Language models were only trained on masking, on unmasking residues in a sequence. So the fact that they have the ability to predict structure is quite remarkable because it's not part of their original training routine, if you want. So I think that those are some of the fundamental differences between how AlphaFold function and is built up and how language models for structure predictions are built up and function.
And that's the reason why you are lagging a bit behind the precision in predicting because you're coming from another approach.
I believe so, yes. I believe also because the system AlphaFold, the whole machine learning part of AlphaFold has been specifically optimized on this input data type, which is evolutionary information and related sequences. So we need to rethink this part, the machine learning part, and to combine it with the language modeling so that we can have an end-to-end system which is better at structure prediction. We haven't done that quite as well as we could, I think, in the community so far. Again, because the system has been around language models have been around for maybe 5 years tops, 3 years really. So I think there's still ways to go.
Another question, how about the predictability of, for example, RNA folding in different media?
Yeah.
So for example, difference in, nothing, vacuum versus plasma, for example, when the different environments.
I assume this is a structure, so it's in the QA maybe. I assume it's a structure prediction task, or
yeah, predictability, prediction of RNA folding.
Okay, yeah.
Depending on the environment as well.
Right. I think RNA folding, I mean, it's the next big thing on the agenda of, if you look at CASP, which is where AlphaFold really make the breakthrough, and what really drove the development of something like AlphaFold was the competition of structure prediction in protein. And I think CASP now has a specific track for RNA folding. I think it's from a dynamics and from a problems perspective, it's a very different folding problem than the protein structure folding problem. So we probably need to think about different solutions from the deep learning perspective. And then when you start with conditioning in, right, of different medium, well, you can make the same argument about proteins. And all of these things are in development. So AlphaFold predicts the static structure of a protein based on X-ray crystallography from PDB. But we know for a fact that proteins can move, right? They have different states, and they may even be restructured in time by heat shock proteins, for instance, right? So I think as we develop, because in the protein space we had a little bit of an advantage because the data collection efforts have started much earlier, we can probably take some of the learnings that we're building right now into how to customize AlphaFold 2, for instance, for different conformations and different conditions, and probably relate that back to RNA prediction. That's my intuition.
I think one last question for now. How are diffusion models used to produce new chemistry?
Yes. I mean, interesting question. So I think it depends on how you define new chemistry. But in the case of the example that I showed earlier, where you have a binding pocket and you just want to predict what could bind to this binding pocket, I mean, what is the chemistry that you're putting there? Well, the model decides. Obviously, the model is a probabilistic model trained on the chemistry that it knows, so it won't go out too much further than what it already believes to be reasonable chemistry to fit into that pocket. But there are efforts in which you can push the models into spaces and into corners of the latent space of these models that aren't quite as obvious as what a person with a lot of domain knowledge would say. Okay, I add some carbon here and some bond there, and then I'm going to have something that binds. So I think we are still constrained by the data sets that we train our models on. So completely new chemistry may be not possible, right, in that sense. But it's still chemistry that is not obvious, maybe from a expert perspective. At least sometimes it's surprising. That's what I've been told.
Thank you very much. We see us at the end of the lunch talk again.
Thank you.
Now we proceed to our second presentation, which is from Applied AI, Europe's largest initiative for the application of leading, trustworthy AI technology with a vision to shape Europe's innovation in AI. And the presentation will be given by two speakers, Dr. Farda Buzara and by Nizara Lingarajaya. So you jointly, or you split your presentation and will show us your information, what you are doing. Some words on the speakers. Fahd has gathered experiences from holding various positions at multiple tech companies, from startups where he had the position of head of AI to large international corporations as a senior member of a large research and development team. And Nizara is an AI engineer at Applied AI with a background on image processing and machine learning. And she worked as a data analyst at the Leibniz Institute and as a student NLP researcher at the FKI. The title is Driving, you see it, AI Impact in Biotech and Life Sciences. So we are curious about your talk.
Yeah, thank you very much, Andreas, for the introduction and for inviting us to today's talk. I hope everybody can hear me right.
Yes.
And can see my screen. Yeah, so yeah, glad to be here. Glad to talk a little bit today about AI impact in biotech and life sciences. So we will talk about sort of set the stage by talking about AI evolution in biotech, and then transition into the practical AI use cases for the sector, where my colleague Nizara will talk about practical use cases that are impacting the industry. And then we will wrap up the presentation with an introduction to Applied AI as a growing player on the market with respect to AI in Munich, Bavaria, in Germany, and Europe in general. Right, so I think the introduction has been already done. So myself, I'm Farda Buzara. I'm Principal AI Program Manager. I basically lead large engagement programs and projects at Applied AI. And my colleague Nizara is an AI engineer. She's close to action, to the implementation, to really making, to anything around data exploration, POC implementation, productization, and really making sure that we engineer products and solutions around AI, and we deploy these products successfully with our partners. And so, as I said, we want to set the stage first by talking about the AI evolution in biotech and why this topic is very important right now. And in general, the evolution of AI in biotech and in the sector is not a recent phenomenon. It's been around for over 4 decades. It's been gaining momentum. And this is an unsurprising development and trajectory in an innovative environment or sector such as biotech. And so while AI has been initially in a support and assisting role in projects, in earlier projects, it transitioned more to be the key enabler and key technology around solutions, where today, if you're talking about, for example, computational drug discovery, it's actually an enabler technology there. And so AI and machine learning in general, and machine learning as a subset of AI that is centered around discovering and studying data, basically, are revolutionizing the approach to approach complex topics and research projects and this is done by harnessing vast amounts of data, as well as leveraging the available compute power and computational resources. And this clearly highlights the AI's pivotal role in developing innovative solutions for different industries within the biotechnology sectors like healthcare, pharmaceutical, bioinformatics, and biotechnologies, and other sectors, for example, agriculture, and so on. But why is this? Why is AI continuously at the heart of discussions? Why is it gaining a lot of interest recently? And not just for biotech industry technologies and industries, but also for other industries on the market. And this can be brought down to 5 major aspects. First of all, the data explosion and the availability of data. I think it's pretty much clear that for AI and machine learning in particular, data is central to enable AI models to learn patterns, to learn insights, and learn structures in this data that enables us to learn mappings between input and output data, and that is capable of then predicting, classifying, generating new information, basically. And so the continuous stream of new data being generated on a daily basis adds more valuable input to these AI models. Together with the democratization of AI, and this is where basically the availability of tools, of cloud services, of resources in general, is lowering the entry barrier for domain experts to be able to leverage the availability of this data, as well as to quickly develop AI solutions, innovative solutions, and train models and deploy them even. So it's possible and it's doable. The maturity of the technology is also a key aspect here, in the sense that we have seen in the past few years a lot of models performing really well. They are very accurate, very robust. They generalize quite well, and they can deal very well with outliers in different domains. For example, we've seen the recent advances in last year and this year in LLMs. And in some cases, or in most cases, outperforms normal algorithms, non-AI algorithms, and in some cases even outperforms human beings, in the sense that it's faster, better, more accurate, more robust, and can handle large amount of input data and sensor data at the same time. And in terms of computational power, I think, as we have seen previously in the presentation from Christian from NVIDIA, the availability of the compute power, whether it's on-premise servers or cloud-based services, or even deployment on edge, so edge devices being able to also run and even train neural networks and AI models, that all speaks for enabling a quick, innovative prototyping and innovation, a generation of innovation and innovative solutions, and enables also it's a key enabler to using this huge amount of data that is being leveraged across multiple industries. And finally, regulatory advancements. Clearly that when talking about AI and AI solutions for safety critical environments, so like for example in healthcare or pharmaceuticals, it's important that there is a framework that enables that is regulating the market, that is making sure that the AI solutions are safe to be deployed, that the customers and the users are protected. And the recent advancement, for example, with respect to the AI-EU Act, speaks for interest and the focus of government agencies and standardization bodies to really address this topic as soon as possible. One interesting aspect we want to point to is that while it's clearly it's clear that AI and machine learning are very relevant for R&D applications, because it's related to innovations there, and for and production in general, to improve and create new products. Our approach is to consider AI and machine learning holistically, so to take a look at the big picture and to take a look at the value chain, where AI has been proven to bring advantages across multiple components of the value chain, thinking about supply chain, distribution, customer service, marketing, and sales. And the idea is that, together with the advantages on the innovation side and on R&D side, deploying AI across the value chain will improve revenue, increase revenues, decrease costs, improve efficiencies, create new markets, open up to new markets, and then create new solutions. And finally, we've seen now the importance of the technology, the maturity of the technology, and the availability of data and compute infrastructure. this also explains the recent increase in terms of the investments. For example, as you can see on the left side, the funding for AI-driven drug discovery companies has been steadily increasing in the past few years, almost exponentially. This also aligns with the with forecasts on the market. For example, in terms of early stage drug development, Morgan Stanley estimating that the market in the next decade is going to grow up to 50 billion in sales, basically. So this shows that the technology is there, the interest on the market is there. Now is the time to really invest in AI, in the sense that to really dig in deep into the use cases, to allocate resources, and not just to experiment, but also to make sure to address all topics with respect to infrastructure, quality management, regulations, and compliance, to make sure that these experiments result in tangible AI products that can be deployed easily. And with that in mind, I will hand over to my colleague Nesara for the second part, where we dive deeper into practical AI use cases.
Hello, everybody. I hope you can hear me. I'm audible? Okay, cool. So the purpose of this is to just give you guys an ideation of the potential AI use cases in biotech, biopharma, or biochemical industries on a whole. So as we see, as Fahad said, it's not just in terms of R&D or production. If you look at the workflow of pharmaceutical industry as a whole, you see application of AI in various different domains, may it be preclinical, clinical, post-production, production, or supply chain optimization. So the idea here is to just introduce you guys for the use cases in general, and not to give you more information about the technical aspects. So going on to the first one. So this is molecule generation and optimization for drug discovery. As discussed before, we can see the advent of it and how much attention it's gaining in the recent years. So ideally, in any traditional or conventional aspect of molecular generation or optimization, it used to be a clinical process where chemists used to do a labor task. Like, it was very laborious, manual labor task, pure chemistry oriented, where every now and then they used to generate or develop molecules which had a good qualitative drug likeliness estimation, check it, and then again, if it doesn't work out, again repeat the process. But with the advent of AI in this domain, we can see that the generation is made much more easier by the use of generative algorithms and models such as GANs, generative adversarial networks, variational autoencoders, diffusion-based models like we saw in the previous presentation. And what do we get out of this? What do we get after applying AI in this domain or in this particular aspect is that we can have basically, if you take FDA approval for a particular drug, it's minimum 12 to 14 years. So the preclinical, which is the R&D process itself, will take like 4 to 6 years. So using AI to generate molecules and test them in this setup will reduce your research and development time drastically by around 60% of it. So also it gives you higher success rate in clinical trials by saving almost at least 25% of the delivery processes. And also it gives you ability to optimize for the particular property that you wish to do. So basically, it's, if you take VAE, for example, it's just latent space optimization. You just have to optimize the particular architecture for a property, property being solubility or toxicity, so on and so forth. And the next use case being used in medical image segmentation. As we see, the challenge here is rather an opportunity for applying AI here is that if you consider CT scans, PET scans, and all that, usually radiologists just go through it and scheme it and then tell us which of the parts are affected and what is the stage of probably the disease that's incurred. Like, medical image segmentation plays a very crucial role in diagnosis and treatment planning of diseases like cancer, cardiovascular disorders, or neurological disorders, as we see over here. So it is, if you rely on radiologists or purely human-based suggestions or decisions, it's extremely time-consuming, and its inter-observer variability is high. So you cannot expect the same sense of decision made over 1,000 reports. So the idea here is to make it consistent, make it precise. So instead of using humans for this, it's better relied on machineries or algorithms that does this, automate this job. So we can apply image segmentation analysis using architectures like UNet or mask RCNN or RCNN. And for medical image segmentation, one of the very first successful algorithms was proposed by University of Freiburg, which is still which still serves as a state of the art, which is a UNet architecture. So as we see, utilization of this could drastically improve the efficiency and also maintain consistency by not relying on inter-observer variability or relying on human decisions. Although they are domain experts, sometimes even they are prone to make mistakes. And as we see in medical terms, people often tend to take second opinions, and sometimes it so happens that you end up taking multiple second opinions. So it's better to automate such kind of processes and have a consistent and precise decision and diagnosis made. And it also helps in scaling the medical diagnosis part for thousands and thousands of reports. And the next use case being using deep learning or artificial intelligence in clinical trials for patient recruitment optimization. As we saw, for one of the examples is COVID. When COVID kicked in, we saw a lot of people affected and wanted medicines, and which was in the process of development. So after the drugs are developed, they are put out for clinical trials, and for clinical trials they have to pick patients, and the patients are not randomly picked, but they are undergone thorough screening of their health records, and then only the ones who are in do or die situations are picked. And usually it's a manual task where you have to undergo multiple reports and previous patient histories. So you can actually make this much more easier and scalable by applying AI for electronic health records. It's a simple NLP task where algorithms like decision trees or ensemble methods learn patterns and features like demographics, patient's demographics, medical history, and biomarkers, which indicates the patient's eligibility. So this actually results in speedier recruitment of patients and also reducing operational costs and enhances the trial diversity, and also accelerates the entire timeline by up to 20%. And it gives you quicker access to the market, which mean that quicker you get the access to the market, more would be your revenue, and you would be the pioneer, and you would have more customers. So the next one being AI-driven metabolic engineering in strain design and molecular strain design and optimization. So this is basically a metabolic engineering use case. This field is it's a multidisciplinary field which applies principles of molecular biology, biochemistry, or genetics to modify the cellular metabolic pathways in organisms or products that are used to enhance or develop the other potential products, such as biofuels, food products, chemicals, or pharmaceutical ingredients. So the challenge here is to identify that optimal genetic modification that is to be done for this particular strain, microbial strain, which enhances the end product's result. So in order to do this, we can apply machine learning techniques for for predicting optimal genetic modifications and metabolic pathways. And sometimes we can also apply feature engineering to understand the relevant features like gene expression, metabolic flux, so on and so forth, which affects the outcome of the end product. So by using this, you can actually increase the time. I mean, you can reduce the time. Instead of just doing this clinically, you can use AI, and you can reduce the amount of time that it takes to do. And you can also have quality in the end products that you get. For example, maybe biofuels or pharmaceutical chemical industry. It enhances the entire process and quickens it up. So the next one here would be in agriculture and to monitor the crops. So basically here we see that agricultural sector is often overlooked. So it's not just chemical or biomedical part, but also agriculture part also falls in pharmaceutical industry, as they use a lot of pesticides and chemicals for the enriched yields. So here we can ideally as we see usually the harvesting the fields that we used to grow crops sometimes uh, scale up to like acres and acres together, like 300-400 acres, which becomes highly impossible for a particular person to, even if they use some kind of a vehicle to go through across the field. It's a very time consuming process and also stressful and straining. So it's always advised to make this a process automated, because if you use techniques like computer vision, amalgamation of the interdisciplinary techniques like IoT, machine learning algorithms like segmentation or detection or classification and tracking tasks alongside with computer vision and IoT field, you can make this solution even easy. So basically it maximizes the yield by just reducing the amount of stress, amount of manual labor that it takes to overlook the entire property, entire crops. So you can actually see how the crops are performing, whether it's dying or whether you're getting good yield, so on and so forth. So as a result, we can have a maximized yield. And also in terms of socialistic notion, it supports farmers for data driven decision making. And I think this should be particularly subsidized. Such kind of implementations should be subsidized on a larger scale. So as we saw, it's some of the use cases where with respect to research and development and in-field, in situ use cases of AI applications. But now we can see the production application of AI. So if you see entire supply chain and the production workflow of a pharmaceutical industry, we can actually see that this is where it is more relevant and mostly needed. So demand volume prediction is when, as we saw, if we take again the example of COVID, we saw that there was extreme scarcity in remdesivir injections. So you can never know when the demand kicks in and when there is no demand at all. So this is an opportunity for AI use cases or machine learning algorithms to actually be the driving force to make these kind of critical situations much easier. So we can use time series forecasting using autoregressive transformer models to capture the trends and nonlinear trends and seasonalities. So this helps to carefully monitor the production demand and also meet the current demand on time. And if there is high demand, it also helps you to have a forecasting, foresee that you might really need a lot of production, and if there's no demand, you can actually scale it down a little. So these are some of the use cases that we can actually apply artificial intelligence in pharmaceutical industries. And I could just quickly rush you over other ones, probably on strategic pharma unit performance assessment. You can actually go through a lot of, we have the slide deck put up, so you can refer them later, but I think we have a time crunch, so I'll just quickly skim through. So you can actually know the strategic pharma unit performance assessment. As we see, we have different units in pharmacy. So we can use regression models, multivariate regression models, to check which of these units perform well and which do not, and which in turn helps us to escalate the ones which are not performing well, and also keep monitoring the ones that are already performing well. So it gives you a notion of how good the entire organization or the subdivisions of the organizations are performing. So we can also see that we can use them in compliance and safety checks, where the medical industry often deals with heavy amount of data that should be checked for compliance with safety. So we can use large language models to do this task for us by fine-tuning and using retrieval augmented generation techniques that can be adopted for this particular specific task. And since this usually used to be a manual process, now we can have high savings and save the manual effort by up to 90% and significantly reduce the manual search time. So these are few of the use cases that we can see in pharmaceutical industry, biopharma, or biomedical industries, where we can actually apply artificial intelligence or deep learning or machine learning techniques on the whole. So moving forward, handing it over to Fahad to continue and on it.
Yeah, thank you, Nisada. Considering the time, I will just jump directly to the very last slide. I want to point out everybody to the long list of white papers that we have on our website, where you have a lot of useful information about technical and non-technical topics with respect to the AI that, in my opinion, very interesting to everybody. Again, for technical and non-technical people. And yeah, feel free to reach out to us. We offer solutions on the traditional consultancy topics with respect to AI strategy and transformation, but we're also heavily involved in engineering and implementing AI and in helping our customers to create AI products out of them. So it's not just about prototyping, it's also about really productizing AI sustainably. Thank you.
So thank you very much for that whole variety of use cases and options we have. And I assume that with all these use cases, you can help and support the implementation in companies or organizations where you want to work with
Exactly. So our model is that we work closely with our partners to and embed ourselves within their organization, where we work closely with the domain experts, and we bring our expertise in AI and data science in general. And we go through multiple phases, as I said, from early exploration phases where we focus on the data, on the availability of data, of do we have the right data to train the AI models and so on. Along the way, then to the POC implementation, really then focus more on the solution development, considering all the requirements that have been defined, and then to productization, where we focus rather on the solution infrastructure, operations, on quality management, and along the way, we also always consider any specific compliance relevant topics, especially for high risk use cases.
And what are the main challenges which are there for the organizations when they want to implement AI?
Yeah, I mean, I would say the traditional challenges with respect to data, so the availability, having sometimes the huge quantity of data might not be even enough, in the sense that you need certain type of data, you need them to be labeled, for example, which creates a lot of overhead in terms of investment and time. And then also like the availability of know-how and knowledge within the organization. So really infrastructure within the organization to manage the topic of AI. And here thinking about large organizations where you have input from different departments and across the entire value chain, and you have to sustainably, know how to identify the top use cases, focus, allocate resources on the ones that are bringing a lot of added value. And then I would also say ethical and regulatory compliance is definitely a challenge in a good way, especially when talking about industries where there is a lot of regulations and a lot of where users need to gain trust and need to really trust that there is an AI component in the solution and they are interacting with. And this is where it's important also to allocate adequate resources for explainability, for transparency for the AI models, and really transparency by design is not just after the model has been developed, such that it's something that is at the core of the design of the solution, basically from the beginning. So just to name a few, basically.
Thank you. Christian, could you show up again? Well, as we are a couple of minutes over time, maybe just one short question at the end. Of course, we will stay, BioM will stay with that topic throughout the year. We will have more events on that, and it will be the main topic on our main event, the new BioConnect in July 2nd and 3rd in Munich. So please try to find out if you can be there. We want to show where it can be used, where it's where there are application right now, but well, short answer on what are the promising future directions of AI in biotech on both. Who will start? Then Christian.
Oh, wow, okay. So I mentioned protein language models. I mentioned nucleotide language models. There are works in the genome language modeling space. I think what we're moving into is many different representations of biological entities. So I briefly mentioned single-cell sequencing. That's another model that actually people in Munich, Fabian Theis at the Helmholtz is looking into. Quite interesting direction. There are many other measurements that we can take at scale of biology. And all of these measurements and put them together into a single model that has access to all of these foundational, let's say, layers. I think that's going to accelerate the way we think about biology. We think about drug discovery. We think about how to, target certain proteins or certain genes. So that's the way I look at it. Yeah.
Bringing together more platforms into one single picture.
I think the usual term here is multimodal models. I'm not entirely sure I like that term too much. It's more about a lot of different foundational models put together into a hierarchical system that has an understanding of processes in biology.
Thank you. Fahd, Nisara?
Maybe Nisara, if you want to take this one.
What was the question again? Can you come by?
The promising future directions for AI in biotech.
So basically, promising future directions would be to actually inculcate explainable AI, so making it trustworthy. So we don't know most of the AI models and deep learning architectures are black box oriented. So we don't know what happens inside of it. We just rely on the output and we believe that whatever output that it's giving is believable and trustable. So explainable AI is the new area and a research topic where we make these decisions that are made by AI models more trustworthy and we make them more interpretable. I think future direction, this is not just in terms of pharma industries, but also on the whole, this should be taken into consideration and we have to make it more interpretable as much as possible. Yeah, this is my take on it.
So many thanks to the speakers for the interesting insights, and of course, many thanks to you all for listening and watching. I hope you will be back next time. See you again.
Thank you very much.
Thank you for having us. Bye-bye.
Bye-bye.